Hi, I'm Jan Witkowski. I'm Director of the Banbury Centre here at Cold Spring Harbour Laboratory. And this is I think the third afternoon of the 80th Cold Spring Harbour Symposium on Quantitative Biology. And the title this year is 21st Genetics, Genes at Work. And I'm delighted to have with me a very old friend of the lab and of me, Arnold Levine from the Institute for Advanced Studies in Princeton. I'm going to talk about the presentation he'll be making
on Sunday morning.
So as a preface, Arnie, you're going to be talking about P53.
That's right.
So how long have you been working on P53?
Well, my lab was one of the two labs that discovered the P53 protein in 1979. So we're approaching our 45th anniversary.
Approaching a half century.
That's right. and we better know something about it by this time. But it turned out to be a very important gene in the origins of human cancer. It's the single most common gene that's mutated, the somatic mutations, in cancers. So that 100% of ovarian cancers have P53 mutations, 70-80% of lung, 75% of colon, all the way down to only 2% in testicular teratocarcinomas. So there are some tumors that really depend upon losing the function of this protein to give rise to a cancer, and other tumors that seem to ignore it.
Well, we might come back to that, but maybe we should stick for the moment with what you're going to be talking about here.
Right. So I thought I would just tell you a little bit about what I don't have enough time to talk about, and that is the human aspects of P53 protein. The P53 gene, when mutated, can be inherited in families. And these families, which are all over the world, have been collected and are watched very carefully because they will develop cancers over their lifetimes.
This is Li-Fraumeni.
This is called the Li-Fraumeni syndrome, right? It's named for Li and Fraumeni, who discovered this syndrome. And the people who inherit the mutation will develop anywhere from 1 to 7 independent cancers over their lifetime. Now, we have a database that has been collected that has roughly 620 families. There are approximately 6,000 individuals in these families, and there are really 30 years of observation on the families. And so we've used this database to analyze the history of this disease. And there are three very interesting things that are properties of these inherited cancers. The first is that they occur at various times during your lifetime. So early on, at ages 0 through 4 years, there are cancers like medulloblastoma, which is a brain cancer, choroid plexus papilloma, that's a brain cancer, adrenal corticocarcinomas, that's an adrenal gland cancer. And this happens early in life, and that's because the cells of these tissues are actively growing at that time. And then from about 5 years old till about 20 years old, one sees osteogenic sarcoma, bone and muscle tumors, rhabdomyosarcomas, and that's because those cells are now taken over and are actively growing. And then between 20 and 45, the most common cancer is breast cancer. And that's because every month breasts expand and contract. And then it filters off, so that by the age of 70, if some individual has never gotten a cancer but has this mutation, the risk of getting a cancer between 70 and 100 years of age is actually lower than the rest of the population.
Yes.
And so that first clue that comes from the age of onset of tumors and the risk of these tumors, which are as much as 100-fold higher than normal, the risk, tells us that stem cells are likely involved in the being surveilled. They're being watched by the P53 gene. And if you have a mutant, you fail to surveil. And then finally, most of these tumors turn out to be tumors of the mesenchyme, a particular germ layer. And it's rare that people with Li-Fraumeni syndrome get endodermal tumors. Endoderm would be lung and colon and stomach and pancreas. Mm-hmm. And yet it's common to get mutations in the somatic diseases, right? So there's something about surveillance of stem cells of mesenchymal tissue from the inherited form of this disease. And that's given us, just the epidemiology of the disease has given us one of our clues as to the function of this protein. It's watching over the stem cell and making sure that it doesn't really make mistakes. And that's one of its really important functions.
So why is it such a rare mutation why is it such a rare hereditary mutation?
Yes, so it could be, it's quite interesting. The frequency of this mutation looks like it's lower than other kinds of mutations in the genome, and that could be for a couple of reasons. One is one could have abortions at very early ages. You might not even know it.
Sure.
So you'd never report that, for example. Or it really could be that since it's involved in stem cell development, these abnormalities that come up mean that people don't live through the whole pregnancy. And so we think that that's a hypothesis. Now, the mouse, which is a very good model for this disease, does in fact have birth defects. There's one birth defect is runting. It's a small size. All the mice will develop tumors. This is in fact what I'll talk about is the mouse at the meeting here. The mice develop tumors. The single most common tumor for the mouse is a thymic lymphoma, which is a blood tumor that comes from the thymus. And the thymus is a place where T cells mature and make receptors. And every T cell has a different receptor. So that's a very interesting thing. It's like a zip code, each T cell. Because that means that if the disease was clonal, that means arose from one cell, the whole leukemia or lymphoma would have the same receptor. Right. But if it's not clonal, but arises many independent times, there would be tumors with many receptors, and it would be very diverse, right?
So is P53 the only mutation that occurs?
Yes.
To begin these lymphomas?
Yes, it is the only mutation in these mice that begins it, but it becomes quite clear that you need additional mutations. So we would say it's necessary for the disease, but not sufficient.
And can you do the subsequent mutation? Do the genes in which subsequent mutations are needed? Do they occur in the same order, or are they the same genes across all these lymphomas?
That's exactly what I'm going to talk about. That's one of the things that we really can measure now. So it's quite remarkable because the mutations occur randomly, but the selection of the mutations occur in a defined order. So the very first mutation is P53, and it results in abnormal genomic stability. So the genome is unstable. It starts to have deletions and additions and make a high mutation rate. And that's the first problem for the cancer. The second is a mutation in a gene called P10. And P10 regulates metabolism, glucose metabolism. So the T cell gets an abnormal metabolism. It takes up too much glucose, and it grows better, right? And that mutation actually occurs before the T cell receptor is inserted. So it's a very early mutation. That's how we know it's so early. It's happening during development. And then the third mutation is in the cell cycle genes. So there's a wonderful set of cell cycle genes that promote growth. That's called cyclin D, CDK4 or 6. And those are both mutated, they're amplified, and that's the third mutation. And then, so we have genomic instability, we have metabolic changes, we have growth of the tumor, and then finally, we have a mutation in the gene that blocks differentiation of the T cells.
So it keeps
it as a like stem-like cell. That's right. So those four mutations occur. They occur in that order, right? They occur randomly, but then they're selected in that order, right? And then what happens is, remember I told you that you could count the number of independent events. So we have as many as 1 transformed cell a day by 9 weeks of mouse's age. So the mouse is 9 weeks old, and you get now one a day, right?
But when you say transformed, fully transformed, all these 4 mutations
Fully transformed with all those mutations growing like a lymphoma.
Wow.
So that means there are as many as 60 different independent clones that arise, but by 20 weeks you only see 1 or 2, which means they compete with each other.
Do you mean literally compete directly with each
That's
other,
right.
or is that...
Well, the reason we can tell they literally compete with each other is by 20 weeks there's just 1 receptor.
Right.
But by 9 weeks there's 5 receptors or 10 receptors.
But what's the nature of the competition?
Right. So the competition is growth, for growth in space and resources, right?
Okay.
So it's just like it's Darwinian, right? They live in the thymus and they crowd out the other cells. They need resources, and they're the best at getting the resources: glucose, other nutrients, fats, carbohydrates, and they get those, and then they grow. So they outgrow their neighbors.
And if you could block any one of these steps...
Yeah. So we're just doing that now.
Can you do... Are there
Yes,
methods
so
to
there are
do
drugs that, since we know the genes now, we have drugs. For the PTEN mutation, there are PI3 kinase inhibitors.
Mm-hmm.
They're called gamma delta PI3 kinase. It's just been approved, actually, for human leukemias and lymphomas. So we're trying that drug. That would be an important drug. And there's a cyclin D CDK2 inhibitor, and we're trying that drug. So we would like to see if we add the drug, at what time we should add the drug, and what will it do? Will it change the pathway, the direction of the pathway, and you'll get a new tumor? Or will it actually be fewer clones that arise? Or will it be that a different clone will arise all the time?
Think about an order of mutations.
Yeah.
I think of Bert Vogelstein
That's
and
correct.
colon cancer.
That's right.
So are your data on these mouse lymphoma
Completely similar to Bert's. The genes are a little bit different. Bert's first gene is in the Wnt pathway. We don't have a gene in the Wnt pathway, right? Bert's second gene is RAS, and RAS is in the PTEN pathway eventually, right? PI3 kinase pathway. So that's in common. Bert's third gene is a TGF beta-like gene. It's a SMAD gene, and we don't have that. But Bert's fourth gene is P53. And that brings us back to the fact that in colon cancer that's spontaneous, P53 is a late mutation. It's the last one to occur. In the inherited case, of course, it's the first one to occur.
So we've only got a couple more minutes, but so briefly, going back to what you said at the very beginning, that the incidence of P53 mutations varies enormously depending on the tumour type.
That's right.
Or presumably originating tissue type.
That's right.
Is there any clear understanding why that should be?
No, we do believe that different signal transduction pathways occur in different tissues. And there's something very special about mesenchymal tissues that are surveilled in some way. What P53 is a transcription factor that responds to stress. But it seems not to respond uniformly across the body to stress, but certain tissues more than others.
Yeah.
The immune system, for instance, is very heavily surveilled.
Yeah.
That's right. Yeah.
Well, Arnie, thank you
Great.
very much.
It's a great pleasure.
Thank you.
Okay.
Yeah,
Great.
fascinating.
Yeah. So